Thank you, Nancy, for that very kind introduction. And to put some perspective on things, I started teaching in 2020. So I could very much sympathize with the last speaker's issues. I often was speaking to a box and going, is this working? Is anybody out there? But so today I will be talking on behalf of myself and my colleague, Allison Stegner. We met when we were both postdocs, and neither of us really studied vegetation up to that point. As the introduction suggested, I studied algae and diatoms, which also have chloroplasts. So it was really cool. It was very awesome to see the poster out there with a circularized chloroplast, because I do a lot of work in genomics now with diatoms. You'll hear a little bit about that on Monday if you come to that talk. And Allison actually studied small mammals, but we met and we were really interested in environmental change. And so we looked for data that we could address that question with. So let's see. Okay, great. Everything's working. We all know we're in this period of really intense change. But what we don't have a really great idea about is if that change is within sort of a normal range. We understand baselines that might go back 100, 200 years. We heard about some soil archives that allowed us to better understand past soil history. But when you get further back in time, we don't have that monitoring data. We don't have those collections very often to give us good context. So that's where paleoecology comes into play. And we can really start to ask whether change is normal, and then hopefully better understand what change might look like in the future. Okay, so this is my favorite tool, paleolimnology. I love aquatic ecosystems. I grew up in Wisconsin, lots of lakes. Not so much when you get down here, but there still are some. And what lakes do is act as a collecting device for all the sediments on the landscape. They flow to that depression, and we have an archive. And there's lots of different things that get collected into that basin, from pollen, which we'll be talking about today, to things that are happening in the lake, within the watershed, groundwater inputs. All of that can act as multiple proxies so that we don't even just understand what's happening maybe in the ecology of the landscape or the ecology of the lake, but we might even be able to scale up to understand some of the drivers, some of the paleoclimate that's happening that might be driving some of the changes we see to the ecology. Okay, so how do we do this? We go collect mud. It's my favorite part of the job. We go out usually to a lake, but sometimes we might be in wetlands or other lowland environments. And we use some sort of device to collect the mud. Here you see a gravity coring device, which is basically a tube with a weight on it. And we use gravity. We just send that through the water column. It pierces that sediment water interface. We get some section of mud. Now, not usually that much, so we have to scale up. And from there, it can get very complicated. There are coring techniques in which, sorry, I'm a wanderer, and I apologize if you're joining on Zoom, but freeze coring uses CO2 or liquid nitrogen and freezes all of that sediment onto a metal surface. This is really great when your sediments have a lot of stratigraphy. Here you can see there are a lot of bands in that sediment that can tell you something about how time was passing and the different environmental conditions as that stratigraphy is laid down. Sometimes you need lots of materials, so you use a multi-core that has multiple tubes. You get more sediments, you can do more analyses. Piston coring, by and large, the data you see today was mostly done through piston coring. Piston coring allows us to go back into a hole over and over again to get pores that might go 8, 10 meters depth instead of the short pores we get from gravity coring. And then if you have sediment that's unconsolidated, we sometimes use vibrocoring, which uses vibration to get through that unconsolidated sediment. Really great for sandy sediment, not so great for organic rich sediment. And then it can really scale up from there. Kohlenberg coring takes 6-10 meters of core at a time with one big giant coring rig. So that's just an idea of the variety of ways in which we get sediment from the bottom of lakes. Then we subsection that sediment. If we're doing the gravity coring, we might just extrude that core in the field where we just push it up a centimeter at a time, scoop that sediment off and put it in a container for later analysis. More often than not, though, we split cores down the center and then discretely sample every 10, 15 centimeters. It really depends on how much core we have to give us a resolution over time. And then we analyze it. This is my favorite analytical technique. I look at microfossils. This is also the analytical technique for pollen analysis. But almost any type of analysis you can do on soils, sediments, water, you can also do on material that you get from the bottom of lakes. And this is what we're after. Here we just have a depth profile of that sediment and some proxy we're looking at. Here are diatoms, that's my favorite proxy. But that could be really anything here. You could put total organic carbon. It could be pollen grains, a species of pollen, almost anything. But depth only tells us part of the story, right? We can say this happened before that, but we really don't know the timing of events. So we have to change that depth into time, and that's a bit tricky. We can use a lot of different methods to date our sediment. Mostly, we use radioisotopes, and primarily lead and radiocarbon. Lead's good for a couple hundred years, radiocarbon further. When you get into really old material, uranium-thorium series is helpful, as well as strontium, but strontium usually in the oceans. Volcanic eruptions are also really helpful at telling us time, because that ash layer can be directly tied to an event. Paleomagnetism, the poles have changed in the past. We can look at magnetic elements in that core material to also help us date. And one that might be relevant to pollen in plants and vegetation, introduced taxa. If we know a species was introduced to an area, that also gives us a really nice tie point. And this becomes really important because we really can't put things into perspective without having that time. The time is always modeled, which will become really important to the story I'm about to tell you. All right, so pollen's awesome. Gives us a lot of information about past vegetation. It's also a little bit biased. It's going to be biased to those taxa that are wind pollinated. They're going to be predominantly what we see in the cores, but there's also some portion of non-wind pollinated taxa. And what we're really after is understanding some change in vegetation. So this is an example from Steel Lake, Minnesota, and we have time here on the x-axis, and on the y-axis is percent woody cover. And you can see that there was a major shift in the ecosystem through time, where you have woody cover giving way to a prairie ecosystem. And the shift was very abrupt, happening about 8,000 years before present. And these are the sorts of things that pollen is really good at telling us, is about these big, massive changes in vegetation. Okay, so I'm going to shift to the reason this work can be done. Pollen has been studied in core records for over 100 years. It has many researchers and many places that pollen records have been taken from. And all of those pollen researchers got together, well, not all of them, a group of them got together and decided we could make better inference if we made a database where we all shared our data. And this has been a movement, I'd say, over the last couple decades in which people are moving towards having data open. I think now it's not really an issue, but when I first started out, it was just being sort of taken up as, hey, let's all share our data so that we can make global scale inferences. And so a group of folks started the Neotoma database. Pollen was what they really focused on to begin with, but Neotoma now holds records for diatoms, for like sedimentation rates, which is really important to understanding, the movement of soil and sediments, and many other things. So each one of those little figures at the bottom of this diagram shows one of the constituent databases in Neotoma. There's also an R package that's associated with it so that you can really easily analyze these data. And if there are teachers in the audience, they also have educational packages so that you can use this in the classroom. Okay, so today I'm going to be talking about a lot of records from the Neotoma database, but I thought it'd be nice to highlight a few that's in the meta-analysis that I did with Allison that are nearby. This is Horseshoe Lake, which is just outside of St. Louis. It's just south of the confluence of the Missouri and the Mississippi. And here is a satellite image of that lake. You can see that this lake is a oxbow lake. These are formed as abandoned channels of meandering streams. There are many different ways lakes are formed. This is just one of the ways. One of the most common ways is sort of glacial processes. That's why there's so many lakes in the north, in Wisconsin and Minnesota and Michigan. Also, tectonic processes can form lakes. Those are some of the oldest and biggest lakes, not really around here so much. As well as dissolution can create lakes. And then we also like to create lakes, right? Impoundments, dams, a lot of lakes around here are formed that way. Okay, so Horseshoe Lake, not very far from here. Here's the pollen record from Horseshoe Lake. So here we have that calibrated radiocarbon years before present. So this lake's only about 3,500 years old. That's fairly common for these oxbow lakes. That's pretty much when that river was in that spot for that period of time. A lot of glacially formed lakes in the north are date to the last glacial period where the retreat of the glaciers left behind scouring, different formations. And so then you see the percent abundances of each one of those pollen through time. And that's about 3,500 years. Now here is another somewhat local example from Neotoma that's part of the analysis I'll show you in a moment. What's different about this is it's not a lake. This is a core that was taken from what is now a farm field. It's a depression in the landscape. And the thing I want to point out about this lake is it doesn't have the same period of time as that oxbow formation. And that's pretty common of a lot of the records in Neotoma. They might not be complete. They might not be all the way from the Pleistocene to now. They might be incomplete for lots of different reasons. And this probably had some erosion, right? It's a field. It's not continuously depositional environment, might have had some erosion, and that's where you lost some of that time. Okay, so now I'm going to shift and change and talk a little bit about abrupt change, because the main question in our research is really, is change now any different than change in the past? And in order to do that, let's contextualize what I mean by change a little bit. You can have no trend over time, so nothing is changing. You can have linear change where increase in temperature has a equal change to vegetation. Or you can have change where you have a little bit of change in the driver, but a massive change in the response variable. And that abrupt change can either be persistent or temporary. And that persistence and temporary aspect about that change has to do with the driver response relationships. So again, that linear change, you have a little bit of change in temp, a little bit of change in the vegetation. But if you have threshold dynamics, you can either change things and the direction you go in, like if you change temp a little bit, you have this big change to a different state. But you can just change temp back a little bit and you change back to that previous state. That is without hysteresis. If you add hysteresis, there are feedbacks that no matter how much you change back, it's persistent in a change state, which we usually call critical transitions. And so in this research, we are looking at both of these abrupt changes with and without hysteresis. And we're asking two questions. On a continental scale, have there been periods of elevated abrupt change over the last 14,000 years? That's our main question. But then we also wanted to do that across a large spatial scale, continental-wide, because local factors can have very local responses in an ecosystem. But if you have many, many ecosystems that are changing at the same time, you know that there is a large global driver. Okay, so I'm going back to Neotoma. At the time we started this analysis, there were about 2,000 fossil pollen records from North America in Neotoma. Not all of those records fit the bill to do the type of analysis we needed. So we had to cull those records based on some criteria. It just needed to have a sufficient number of samples in order for us to run the analyses for looking at changes. It also needed a duration, a period of time, a minimum number of years to analyze. And then the resolution had to be robust enough that it made sense to apply the methods that we're going to apply. And then we really wanted robust estimations of ages, because if we are analyzing data and events from one area, we have to have a really good estimation of that age to compare to events that are happening across the country. Okay, so we used work of a previous scientist who did all of the age models in a Bayesian framework, which was really, really helpful. And so let's take a quick look at what these age models look like. Here you see on the x-axis, we have calendar year before present, and depth is plotted on that y-axis. And each one of those black tick marks is a tie point. We don't have ages for every single centimeter of the core. We only have a few, and those give us a tie point. And when we model age, you could put a linear model up here and just run a straight line through everything. But the Bayesian age models allow us to fit a monotonic line through the tick points. The Bayesian age models also allow us to repeat that process over and over again to build a 95% confidence interval for our age model. And that confidence interval gives us some better estimations of events that happen through time. And that's the power of these Bayesian age models. Okay, so our analytical framework is that we identify abrupt vegetation change for each of our sites in our data set, and then we find the number of abrupt changes across all of those sites. All right, so we resample that age model. So here's that red highlight through that is just one sampling. And why it's so important, I want to underline this fact, why resampling matters so much, is that a lot of times paleoecologists will use the mean of the age model, but that can give you wildly mistaken ages of events, and then they're very hard to compare across space. Okay, then we look at abrupt changes. So I gave you the examples from around here of these pollen abundances through time. And this, again, is that example from Steel Lake, the one that had the woody cover shift. And you can see the major changes in this pollen abundance diagram. You can visualize them. They kind of have big breaks here, big breaks here. We can say, hey, this looks like it has about 3 changes, but we want to formalize this. We want to automate it. We want to use a technique to find those abrupt changes through time. So we use Bayesian change point analysis. This BCP is really good at estimating the probability of a change in both the mean and the variance. It does this automatically. It does have some drawbacks, however. It's sample rather than time-based, and it detects abrupt shifts regardless of whether or not they're persistent. So, and why does that matter? So it will put the same weight on a shift that would be like from wildfire, where regrowth is very quick, or something that's a progressive change that's long-lasting, like changing from the interglacial to the glacial. So it doesn't really understand time. Okay, so we iterate this process for every single data set in our selected data. And when we use Bayesian change point analysis, it detected 5 shifts in our data set, but it also gave us confidence intervals around those points in time. Okay. This has a lot of stats in it, so I hope you're all with me so far. Basically, picked some sites, then we found some change points, and we have robust models. But what we had to do to time is put it into bins. So these are 250-year bins from 14,000 years ago until present. And then we looked at the number of samples per bin. And you see this trend. There are not very many samples, so those are sites 14,000 years ago, and there's lots and lots of sites more recently. This is a problem, right? Because if you have more sites, you're going to have more opportunity for abrupt change. So we only sample the fixed number of sites per time bin so that we give every time bin a fair chance at having abrupt changes. Okay. And here's an idea of what that output looks like. This is Abalone Rocks, which is a site. And so these are 3 different change points. And then these are each of the estimates of each one of those change points. So you can see that those estimates change quite a bit, 3,931 years before present, compared with 4,649 years before present. That gives us that confidence interval, and we can have a more robust estimation of when that change happened. Okay. And then we iterate this a bunch of times, and we calculate those confidence intervals. And here are the results. After all of that, you get one graph. All right, so what you're looking at here are those changes through time, 14,000 years ago until present. Oh, pointer. So we see this elevation in abrupt change about, at that shift from the Pleistocene to the Holocene. This makes a lot of sense, thinking of moving from glacial to an interglacial world. And then we have this giant uptick here, starting about 2,000 years ago, but really takes off about 250 years ago. All right, so mapping the Pleistocene and the Holocene onto this graph. And then, I don't know if you've all been following the sort of debate about the Anthropocene, but they decided not to ratify it. And, we could have a conversation about that later. It's probably because, where does it go? I don't know. They said 1950, they had good reasons, but it's probably because a lot of scientists have evidence like this where we're like, it's definitely the hockey stick, but where does that start? We have to take that up again in another 10 years when they'll allow us to potentially name the Anthropocene again. Okay, so it is worth noting that they did subdivide the Holocene recently into different periods. We didn't see any, and the idea of subdividing these is that there were big climate changes at 8.2 and 4.2, but we didn't really see those in our data set, at least not as much as we saw this Pleistocene to Holocene transition and then this big hockey stick in the most recent past. All right. So yes, vegetation has abruptly changed more frequently over the last 250 years than any other time since the end of the Pleistocene. And increases in abrupt changes have been more rapid over the last 250 years than any other time. And what do I mean by more rapid? This uptick is pretty gradual. That uptick is extreme. All right. Get done, and then you're like, well, we came up with this method. Let's ask a couple more questions, right? We came up with this sampling design. What about biodiversity? What about ways that communities might be changing, driven by anthropogenic pressures, like species removal, invasive species, agricultural species, migration? all of those things might start having some impact on these species compositions. And so we decided to kind of look at our abrupt change and compare it to biodiversity metrics, but then also this idea of species loss and species gain. And basically what we were doing is taking, relative to the last 1,000 years, how many taxa in a sample have been lost, and then similarly, relative to the last 1,000 years, how many taxa in a sample have not been seen previously. Okay, so we did that, and then we layered one other thing on top of our sort of trying to understand biodiversity change. And that's this idea of first appearance datums and last appearance datums. So instead of this idea of loss and gain, it's like, when do we see a species for the first time, and when do we see a species for the last time? These are really important metrics for delineating epochs when you're like thinking about geologic time. Very frequently, when there's a mass extinction, that'll show up in last occurrence datums. But when there's a mass radiation, that'll show up with first occurrence datums. All right. So here's the big graph from our paper. We've already talked about abrupt changes, sort of that big peak at the Pleistocene-Holocene transition, and then most recently. Then SQS. This is a measure of biodiversity, richness. I'm not going to go into the details of how this particular richness is different than every other flavor of richness, but if you want to talk about that, find me. But if you were to be looking just at this metric, you'd think things were fine. Richness looks great. It's increasing. All right, species loss and gain. We have similar sorts of patterns going on where, the Pleistocene-Holocene transition, we have, elevated loss and some elevated gain. That makes some sense. And then more recently, we have an uptick in loss and a decrease in gain in species. So that's pretty interesting. And then the fads and lads. So at the end Pleistocene, elevated fads, first occurrence datums, that seems to make a lot of sense. As deglaciation is occurring, there is more opportunity for first appearances of taxa. Then we have, over the last about 1,000 years, an uptick in lads, so those last occurrence datums, so extirpation occurring or migration happening. And then a more recent uptick in first occurrence datums. We interpret this as possibly migration or potentially introduced species. Okay. And we said, oh, well, that's fun. But what about various regions? Are there places where there's hotspots of losses, hotspots of gains, more abrupt change than in other places? So we subdivided our data by ecoregions. Each one of the colored ecoregions had enough data to be resampled. Fortunately, the center of the country didn't have enough data to run these analyses. And I'll just run through each one of the ecoregions really quickly to give you an idea about how things are changing as you sort of move north to south and east to west. So this is the first region. This is tundra taiga. I guess one of the most notable things in this graph is it doesn't go all the way back to 14,000 years before present, but that makes some sense because of the glaciers. It also has really massive abrupt changes about 8,000 years ago. So maybe we are getting some of that 8.2 event or one of those subdivisions of the Holocene. Pretty flat richness. And then this somewhat big increase in last occurrence datums in that data set. Okay, moving a little bit further south in the northern forests, we have elevated abrupt changes at that Pleistocene-Holocene transition, that U-shaped richness curve, and then, pretty much opposite trend in gains. Those kind of decrease through time and loss increases through time. And then we have lots of first appearance datums in the beginning of the record, and then a lot of last occurrence datums at the end of the record. Okay, moving on to the Western forested mountains. Pretty flat overall. We do a little arm waving here. Maybe this is a little bit more buffered from massive change. Maybe there's more resilience in a lot of these vegetated areas. These regions, all of these graphs I'm showing you are really rife for additional studies because we didn't really spend much time with it aside from here are some overarching trends. And then when we get to the eastern temperate forests, we see something that looks a lot like our continental scale trends. This might be driving a good portion of the signal we're seeing, even more drastic than the whole continent. So I think that this portion of the country has probably the most responsive taxon. So then we really wanted to take all of that data and see what other drivers are potentials for what's going on. So what you see here in this graph at the top is temperature anomaly, that sort of hockey stick, as well as we plotted out changes in percent land cover, focusing on cropland, rangeland, and urban areas, which really increased over the last 250 years, plotted over top with human population, and then some of our more motivating graphs here on abrupt change, and then our fads and lads of the last couple thousand years. Really tying human change to the landscape change. Okay, so just to conclude, I hope I've motivated everybody to think about paleoecological records as useful archives for understanding human change. And I think one of the things that I took away from this project the most is that richness can sometimes be deceiving. And looking at other metrics of vegetation change or biodiversity change in general is really important to better understand impacts of humans on the planet. And not all ecological changes that are important are rapid, but we are living in a time of accelerated change. So understanding those types of changes, I think is very important. Okay, so I went through that rather quickly. I'll definitely take any questions that you have, but I really want to acknowledge all of the people who have contributed to Neotoma. This work could not have been done without all the efforts of those people who gathered thousands of records, and just the community that has been so generous with their scientific research. And really anybody who shares data that's so appreciated to the community. So thanks.